Hello. So good morning, good afternoon, good evening from wherever you're joining us in whatever corner of the world. On behalf of SONA, I am happy to welcome you to the SONA Webinar Series for the year 2026. We are very grateful to have you creating time for this and to join us from different parts of Africa and also beyond. Thank you so much. And by way of introduction, I'm Dr. Royhaan Folarin. I'm an assistant professor of anatomy at the University of Global Health Equity based in Rwanda. And I currently serve as the treasurer of SONA and also the coordinator for the SONA Webinar Series. So it's a pleasure for me to serve as your host and moderator today. For those joining or hearing about SONA for the first time, it stands for the Society of Neuroscientists of Africa. So it's a continental-wide, non-governmental neuroscientific organization with core mission to promote neuroscience research, to support education and training, and also foster collaboration across Africa. So by default, the SONA community therefore includes researchers, clinicians, educators, students, everyone who is an enthusiast about the knowledge of neuroscience, because our goal is simply to strengthen the neuroscience capacity across the continent. So one of the initiatives through which SONA advances this mission is through this webinar series, the purpose of which is to serve as a platform for knowledge exchange. And for the 2026 SONA Webinar Series, we are particularly taking an approach of alternation of sessions between scientific presentations and career development conversations in order to give benefits of exposure to new sciences and also give guidance on neuroscience careers simultaneously. So we are being intentional about diversity of speakers across different regions of Africa and also across different neuroscience disciplines. Our goal for this is to showcase the breadth of African neuroscience research and also encourage cross-institutional collaboration. So as a webinar for today, it's my pleasure to have joining us today Dr. Imam Aminu. Dr. Imam, how are you doing?
I'm fine, you?
We are very glad to have you today. Dr. Imam is affiliated with the University of Ilorin in Nigeria, where he leads the Neurotox group at the Department of Anatomy in the University of Ilorin. His research group focuses on investigating neurotoxicity, as you can clearly make out of the topic for today's presentation. And they also look into seizure-related brain injury, and of course, not limited to that. It also goes to include neural repair mechanisms. So his talk today, for this reason, is centered upon the central role of glia, what role it plays from the point we have a seizure-toxicant injury introduced or induced up to the point of us having remyelination. So you can expect that through this study, Dr. Imam will be informing us a lot about brain injury responses, the roles of glial cells. If you want to know more about mechanisms of remyelination or have some insight into neural repair, you're in the right place. So just before handing over to Dr. Imam, I would like to encourage everyone. First of all, Dr. Imam will be speaking for about 40 minutes, after which we'll go into the Q&A session, during which we will take as much of questions as we can. So be sure to start posting your question into the chat section so that we can have the robust engagement as soon as possible, because questions by default will be taken in the order in which they are received. And of course, at the end of the presentation, you will have opportunity to give us more feedback on We'd like to have your feedbacks as much as possible, and there will be a feedback form that will be posted, especially to those who registered for this for the webinar, so that we can always hear more voices and improve as we tag along. So, without further ado, Dr. Imam is right here, and I hope you're happy to take it away from here. Over to you, Dr. Imam.
Yeah, thank you so much, Dr. Royhaan, for that introduction. So first, I would like to appreciate SONA for the platform, and then for the opportunity to speak about what we do in the Neurotox group at the University of Ilorin, Nigeria. Just like Dr. Royhaan said, throughout this lecture, we will kind of take us through a journey of what toxicants can do, especially in this case, insecticides, and how this insecticide can drive the development or progression of neurodegeneration or even affect treatment outcome. So for this talk, we'll be focusing on seizure-toxicant comorbidity and the demyelination and remyelination effect. And then we'll be looking at what some glial cells, the role some glial cells play. So as a guide, I'll be following these objectives in this talk, right? So which will basically to understand the crisis, right? And then identify the comorbidity link, and then investigate the role of these glial cells, and also evaluate the therapeutic pathways that we can actually use to encourage remyelination, right? Then some advice to the African community. Epilepsy is a neurological disorder that is characterized by unprovoked seizure, and it has been said to affect about 50 million people worldwide, with most of the incidences found in the developing world, more specifically Africa. And of recent, there is an increase in the incidences of epilepsy, and the cause of this increase has been widely unknown. And also complicating this increase is also the need for agricultural produce, increasing agricultural produce, which kind of push the population towards the use of chemicals to actually boost production, also prevent insect. There is what I call silent comorbidities of epilepsy in Africa, right? Like I said, the number is increasing. There's an increasing number of incidences of epilepsy in Africa, right? And the gap is actually, the incidence is so high that a report in 2019 reports about 94.4% in some country, where you have 70% in some other country. And this increase in number of incidences of epilepsy is further aggravated by the treatment gap. And we all know, as we all know, the issues in Africa, that most of this gap is not unrelated to the economic exclusion. Most of the epileptic patients could not afford the treatment opportunity, treatments that are available. In fact, what a study we did before coming into white matter or even epilepsy was to ask what drug do people use in the localities, and we found out that people kind of settle for the cheapest drug, or rather they go for alternative therapy, which most time we do not know their mechanisms, right? We are not sure if they're actually working, right? So to further complicate this is the use of insecticide, the indiscriminate use of insecticide. And we use insecticide in control of parasites, in control of malaria, in control of mosquitoes, right? In prevention of pest attack to our food. And then as we go on, we'll show us how much people use insecticide for what can be said from now to be unconventional use, right? So like using it to even preserve fruits, right? To preserve smoked food that we take directly, right? So which means our exposure to insecticide is not only through the farm or farm produce, right? We kind of unintentionally consume insecticide through the food. The primary underlying mechanisms of epilepsy is basically of three pathogenic triad or pathways: GABAergic like an excitatory and inhibitory imbalance, right? Which means the first thing that occurs is that there is an impairment to the regulation of the excitatory inhibitory balance. And then there is also a complication of oxidative stress and inflammation, which result from the complication of the excitotoxicity that is induced by the hyperdepolarization, right? So from this, what are the burdens of indiscriminate pesticide use in Africa, right? Throughout the news outlets in Nigeria or even beyond, when there are cases of poisoning, when there are cases of death, or they call them mysterious death, right? Most of the findings afterward shows that the cause of those deaths are actually exposure to insecticide. And what is more disturbing is that Africa contributes to only 3% of the chemical production, especially any chemical that affects us, agriculture, insecticide, and the rest. Right, we only produce about 3%. However, the 70% of the burden of this insecticide, right, resides in Africa. And there are reports that they contribute to death, they contribute to the growth of disease, they contribute to neurological consequences. So in our group, the first thing we did in 2016 when we started our journey with insecticide was a kind of to expose rats to what we learned from the farmers or what we learned from the vendors of the most used insecticide in Nigeria. And we found out that organophosphates are the most used, right? So, and then we went through various studies to study diclovers induced neurological consequences, then chlorpyrifos, and then we did all this over the years. However, I was speaking with someone in Morocco in 2019, and then we were talking about epilepsy, and then they were talking about the growth of epilepsy in Africa and the idiopaticness of the cause, right? That we don't know what is causing them, right? Then I thought of why I started studying insecticide. Then I asked myself, right, there are other insecticides that are used. I know that organophosphates are actually irreversible acetylcholinesterase inhibitors, right? So I understand that they will affect the cholinergic system, right? And the cholinergic system also have a role to play in schizogenesis. However, there are some other insecticides that have direct link to the pathophysiology of epilepsy. So we started to work on pyrethroids. So we chose two most used pyrethroids in our community, and that is alpha-sialotrine and then cypermethrin. And when we studied the two in male and female, we found that the effect of cypermethrin on most of the markers that are very related to pathogenesis of epilepsy, including membrane integrity, including neurotransmitter system, GABAergic integrity, we found that cypermethrin affects these structures more than lambda-sialotrine. So we asked our first question, right? And this first question was thrown to a master student, right? That is Damilola, right? That is Modeste. What the question we asked was, what are the epileptogenic and cognitive effects of pyrethroid exposure in epilepsy? And this is not something that just came out of mind. It's something I saw in Cure Epilepsy, where they say pesticide exposure increases the risk of all seizure disorders, right, especially epilepsy. So what he did was to expose rats, right, to expose naive rats to cypermethrin and then pentylene tetrazole. What is the mechanistic basic of this? We know that pentylene tetrazole will induce seizure by blocking the chloride influx, right? And we all know that the mechanism of action of cypermethrin, just like every other pyrethroid, is that they keep the sodium-gated ion open for a long time so that there will be continuous sodium influx. So in this, we were sure that the mechanism match each other, right? And this is one thing that I want to kind of remind African scientists when we plan our research, we should kind of look at the mechanism. And this is so that we will be able to explain whatever we find. And what we found was not surprising, that seizure scores was more higher in animals that are exposed to PTZ and CPM. But the question is, do we have cognitive related impairment in this. And why do we ask this question? Because it sounds like, so is cognition also a marker or a symptom of epilepsy? But we know of recent from clinical studies that not only is cognition a symptom of chronic epilepsy, it's also a symptom of antiepileptic drug, especially phenobarbital and the rest, right? So we check for the cognitive consequences of this combination, and we found out that in the T maze, Y maze, and Morris-Water maze tests, we found out that there's actually impaired cognition. But the question is, this cognitive impairment that you see, are they more related to the loss of gray or white matter? So Mudathir, actually in his work, he worked on the hippocampus, and then we saw some neurodegenerative effects in the hippocampus. However, we were not as convinced to exactly which structure these things are affecting. So I saw this paper where the Enigma study, in the Enigma study groups, and I saw a lot of results where they say that more than gray matter, the white matter loss is more related to cognitive dysfunction, right? In the patient, in several patients in several epileptic patients. So we think we hypothesize that white matter alteration may underlie the seizure and cognitive comorbidity that we saw in our first study, right? So the next thing we then asked was, what happens to white matter in epileptic insecticide comorbid brain, right? So what Damilola did was a kind of to do the same thing as Mudathir did, repeat the procedure, but use immunohistochemistry expression for some specific proteins that are related to both the integrity of white matter, maintenance of white matter, and survival of white matter, right? So she expressed for myelin-based protein, which is an integral protein that is in the white matter, and then OLIG2, which is a marker for oligodendrocyte progenitor cells, and then the GFAP and EBA1, which are for the glial cells. And the first finding that she kind of reported, and which we published recently, right, was, and we can see obviously that there's a lot of white matter in these animals, and specifically there's a lot of myelin-based protein, and this can be extrapolated to be a loss of white matter, because we know that myelin-based protein is actually responsible for maintaining the integrity of white matter. But the question is this a consequential effect of the sapamitrine PTZ, or PTZ sapamitrine, like, caused an havoc somewhere that came to affect the integrity of the white matter? So I would like us to kind of revise the role of Glia in actually myelination, right? What we know is that astrocytes are those cells that actually kind of stimulate or recruit oligodendrocyte progenitor cells, right? So they stimulate the recruitment of oligodendrocyte progenitor cells to become submyelinating cells or pre-myelinating cells, and then become mature cells, right? Also, astrocytes also produce what we call trophic factors, right? Even after myelination, these trophic factors, right, support the oligodendrocytes, right, and keep the OPC recruitment steady, so that when there's any damage, there is the readiness of the recruitment of oligodendrocyte progenitor cells. So there are always progenitor cells that will become mature cells that will produce myelin that will wrap. But what does microglia do? Microglia are actually phagocytotic cells, right? They are the primary immune cells of the central nervous system. So they're there to actually remove the defective sheet, so that when there will be remyelination will occur on a smooth axon, right? And we all know what the role of myelin is, right? So after knowing this, what is now the role of Glia in demyelination? Right? We know the role in myelination. We know that astrocyte is very important if there will be myelination. In fact, it is also very important if there will be unhealthy myelin, right? So, but what is demyelination? Demyelination is characterized by damage to either the myelin sheath or the oligodendrocyte themselves. So when there's damage to myelin sheath, right, it is easy to actually recruit oligodendrocyte progenitor cells to produce myelin, to replace the lost myelin, right? But when there is death of oligodendrocyte, right, that is more difficult. It is more difficult to actually remyelinate that kind of exons. And what we've seen play out, if you look at the image on the left, right, you will see that we have population of the oligodendrocytes reduced drastically, and that shows demyelination. But what you see in response to that is an increase in glial cells, and this is going to be called activation of the glial cells, right? So we know that activation of glial can either be, right, in hypertrophy or hyperplasia. So it's either you have an increased number of cells that are mobilized to the region, or you have an increased size. And we should note from here that, because the question will be that, how do astrocytes encourage myelination and also affect demyelination or contribute to demyelination, right? There's what we call LD and pathogenic activation of glial cells, right? And when there's LD, just like, let's take an example in microglia. There are two types of activation of microglia. There's M1, M2, right? So this is a successive activation, right? But when there's a signal for pathology or inflammation, M1 is activated. So M1 activation is a signal that there's a problem, right? And if that is sustained, if it does not become LM2, does not become protective, then it means that is a continuous pathogenic activation. However, if this become M2, then this means that the signal have been received, the body has responded, and then there is now signal for repair, right? So since knowing this, Damilola express oligodendrocyte progenitor cells, right? Like OLI2 in the white matter, then express GFAP and IBA1. And what we saw is very close to what we know from the literature, right? That when there is damage, right, will you expect that the signaling glial cells will mobilize to this region, right? So there will be more glial cells. However, it's not just about the number, it's about also the phenotype of the cells that mobilize, that are seen in the region. And what we see is that the population of the oligodendrocyte of OLI2 positive cells reduced drastically. So we can say that the myelin loss that we saw in the loss of myelin-based protein is not just about myelin-based protein loss, right? It also has something to do with the myelin-producing cells dying, right? What caused this death is what we'll assess later, right? Which Mudathir did later, as part of his master's program, right? So Damilola also found that both astrocytes and microglia are reactive to actually the change that occur in the myelin. So from here we can say that myelin loss, right? Myelin loss, we can, just like I argue, that glial cells are signaling molecules, basically. They're not damaged cells. So they're not cells that come to the brain to damage cells so that we see. They are cells that actually show us the signal by producing cytokines, chemokines that we use to know that there is actually a problem. So, but from here we can say that the myelin loss that we saw in the comorbidity is both related to the loss of oligodendrocyte cells, and this loss is also signaled by astrogliosis and microglia activation, right? So the question is, this glial activation that we saw, right, are they as a result of neuroinflammation or oxidative stress, right? So why did we ask this question, right? When there is hyperdepolarization within the cell, right, what you expect are in succession that there will be excitotoxicity, right? If there is excitotoxicity, there is also, because in our previous studies we've expressed sodium potassium ATPase, which is a pump that we know that is central to the pump of sodium, and we saw that drastically it was reduced significantly in the cypermethrin exposed rat, right? So we from here we know that probably it has something to do with membrane integrity, impaired membrane integrity, right? And also induced inflammation. So in Mudathir work, when from a soup of the brain, so we did ELISA, and then we kind of quantify for TNF-alpha, which is a global inflammatory marker. And we saw that across the exposure, either pentyletrazole, cypermethrin, or the combination, there's an increase, right, in the expression of TNF-alpha, right? And also, as expected, we saw that the membrane, the integrity of membrane is actually affected, is severe, because we saw a leak, right, a leakage in the membrane by the presence of an increasing malondialdehyde, right? However, what is surprising, right, what may be basically surprising is that we now saw that in this same group that there is increased superoxide dismutase, right, which is an antioxidant marker. But however, mechanistically, this is also explainable. The body does not just leave toxicity to continue to occur. The body does not just leave damage to continue to occur to itself. Right? There is what you call the body physiological repair. And this, we think that the level of this SOD is actually a signal, is actually is actually taking advantage of what we call the antioxidant reserve. So the antioxidant reserve is what we see this SOD coming from. So we can say from here that there is myelin basic protein loss in the comorbid rat. There is also glia activation, which we think are convinced that contribute to this loss and sustain that loss. And then we now know that those activations, right, are actually driven by signals of inflammation and oxidative stress. And we all know that oxidative stress, like I said, sometimes oxidative stress is inflammation. Inflammation is oxidative stress. And the reason I said is because reactive oxygen species production, abundance, drives inflammatory responses, right? Inflammation, most times, lead to mitochondrial dysfunction. And this is a kind of vice versa, right? So it is now important because knowing that there is damage, knowing that a substance can cause damage, knowing that when you are exposed to it can cause a kind of more damage, can aggravate the damage that we will see, right? It's not enough, especially in our setting. So we're not here to just study what the glial cells will do, right? But the question is, can we a kind of reverse this process, right? We know that there are AEDs, right? There are anti-epileptic drugs, right? But the major problem is that they focus on the GABA glutamate imbalance or excitatory inhibitor imbalance. So let's not call it GABA glutamate imbalance, excitatory inhibitor imbalance, right? And most of the drugs can either kind of promote GABA or reduce glutamate activities. And we think that probably these are some of the reasons why there's actually resistance to these drugs, or not that there are resistance. These drugs are not sufficient to treat the consequences of a prolonged seizure, and also a seizure that includes comorbidity with other exposures like toxicant exposure, infections, and the rest, and a host of others. So we check specifically vitamin E, and then we ask if we kind of co-treat a comorbid model, right, with vitamin E and a non-anti-epileptic drug. In this case, we chose valproic acid, and not because we chose it because it is used primarily, right, in the first line of treatment, right? So in this case, we re-examine what remyelination is. We know that remyelination is replacing the loss of myelin sheath, right, or oligonucleotide. So how is myelin sheath that is lost replaced, right? Basically, we know that reactive microglia, right, we go a kind of to engulf the debris left from the demyelination, and then from the production from the reactive microglia, there are signals to the astrocyte to be activated. However, like we said, astrocytes produce two things. They produce regenerative factors, they also produce inhibitory factors, right? These factors are beneficial, just like we said before, that they produce trophic factor, and those trophic factors maintain the health, right, of myelin. And then what we see is that once astrocytes actually mobilize oligodendrocyte progenitor cells, right? These progenitor cells get recruited right towards the region, and then they proliferate and move towards the region of damage, and they differentiate along the way. And this differentiation is actually enabled by the production that by the proteins produced by reactive astrocytes. And also, these cells become mature, and then they have their feet on the exons where there is the need for remyelination, right? So the later part of Damilola's work, right, was actually in the second phase was a kind of to treat this comorbid rat with vitamin E and vaporic acid, right? So we do not have 100% recovery, right? Because there are some data that are not here, right? We have a kind of negative result in some cases. However, when it comes to the white matter, we saw that there's actually protection of the white matter, maintenance of the expression of the myelin-based protein. And from here, we can kind of extrapolate that there's actually preservation of the integrity of the white matter, specifically here in the corpus callosum, right? So, and just like we said that there is Glia activation in the model, and then we term that pathologic, right? So to prevent myelin integrity, we think that there's a need to reduce pathologic Glia activation, right? And we can say that from here because we saw that the population in the group that were, and this is how the treatment worked, right? So these animals were exposed to vaporic acid, then they were later exposed to vitamin E, and then they were exposed to PTZ, and this CPM and PTZ. And in this case, what we're trying to do is to prevent demyelination, not reverse myelination. And what has happened here also is that the integrity of the oligodendral cells has been preserved, right? So there is no auxiliary stress in those cells. The cells did not die, right? So because the population of the cells did not reduce significantly, so when you compare that to control. However, when you compare it to the comorbid group, there's actually more healthy cells, more healthy oligodendral cells in the brains of the rat that are exposed to that. And then when we look at the astrocytes and microglia, we saw that there's a reduced population of activated astrocyte and then activated microglia. So we can confidently say that preventing glia activation, especially pathologic glia activation, is essential to preserve myelin integrity, right? So next is, so when we finished that study and then we published the study, right, then a new student came in, and then she was also interested in the project. And the question we wanted to ask, because we see that when we talk about white matter, everybody think of the large white matter bundles, the callosa white matter, the projection white matter, the association white matter. However, we forget that what you call gray matter is also abundant cells, right? And a small population of glia cells and white matter fibers, right? So she was interested in characterizing the white matter integrity in several brain regions. And then we chose some regions that are specific to the symptoms that we see in epileptic patients that they don't like. And what are those symptoms? Fear, anxiety. Right? They don't know when the episode will come. They don't know when they will convulse. Right? So because of that, there is anxiety in them. Right? They don't know. And then because there is repeated seizure in the brain, there's repeated hyperdepolarization in the brain. Right? It may affect memory in some patients. And we also know that seizure is a motor disorder, majorly. Is a motor, symptomatical, primarily, seizure is a motor disorder. So we chose the frontal cortex and the cerebellum. And that is what, but we were trying to be certain, right, of our decision to kind of go through that. As we all know, like that funding is something like that is very difficult in this part of the world, right? So before you spend money, before you use antibody, before you start cutting brain, you must be certain that what you're looking for, right, is ideologically or mechanistically sound, right, is pathophysiologically sound, right? So the Enigma Epilepsy Working Group study, right, showed extensively, right, through their imaging studies that there is significant reduction, right, of loss of white matter in several white matter structures in the brain, right? So the question is, do we also have the same thing in our model, especially in the comorbid model, right? So Chris Oloede, right, then decided to express myelin-based protein in various brain regions, right? And we were not surprised. The only exception to white matter loss, right, which is surprising to us and which probably we're going to repeat, is the cerebellum, right? So we saw that across the frontal cortex, the amygdala, the dentate gyrus, we saw that a comparative loss of myelin-based protein, right, which means the loss of white matter is not only restricted to the large white matter structure. And this is one thing that I see that is popular in the white matter research community. Everybody focus on the corpus callosum, the corpus callosum, the corpus callosum, right? So I think it is high time that we see that is white matter. We know that white matter contributes to information distribution and all this. And we know that it's the white matter that we're talking about. When we say white matter contributes to this information transfer, it's not just about the bundles. It is about what it kind of radiates out of the bundles, right, to all these regions, right? So, and what we see significantly is that across all the regions that we check, there's actually a loss of the myelin-based protein. And then she was also interested to know, just like we see in the major white matter bundle, that there is actually oligonucleotide loss, right? So she was also interested to check if this oligonucleotide white is restricted to the bundle or is across the brain. And then we saw significantly that the number of the OLIP2 positive cells, right, reduced in the comorbid brain. And in the groups that receive vitamin E and the rest, right, there is actually preservation of this. And this is not surprising. Just like we said, pathological glia activation is driven by inflammation, oxidative stress. The same way neuroprotective glia activation, right, is actually encouraged by reduction of those inflammation and oxidative stress, right? So in this case, when vitamin E is able to kind of promote remyelination or prevent demyelination, it's actually not a kind of surprising. So here we are able to kind of take us through like a simple study, what we call a simple study, because it's just like two objective study in our group, right? And we're able to show that glial cells, right, are the primary mediators of myelination, right? This is not new. We're not the first saying this. We're only saying that we're only kind of reiterating what is known. However, what we may be the first contribution to is that comorbid epilepsy and insecticide poisoning, right, can worsen white matter loss, right? And then it's not just that it can worsen white matter loss, it's worsen white matter loss to worsen seizure, right? And we think antioxidants can actually be of rescue if we kind of study them appropriately. And lastly, what you will notice is that we kind of limited our studies to what benefits our immediate environment. And our first question right from start was, what does insecticide, what is insecticide doing in our community? We know that when you buy banana these days, they don't easily get rotten. We know that when you buy fish and you keep insects, they kind of avoid them. And when you go to the marketplace and you think that, there should be fly around, right? I'm sorry to say that. Flies, everything run away from this. And when studies are carried out, when surveys are carried out, these vendors actually say they use insecticides to prevent flies. They use insecticides to preserve the food. However, what they don't know, probably they do not have the knowledge, is that they worsen the case of innocent people who already may have had probably genetic neurological disorders or acquired neurological disorders, right? They worsen their case. And not only that they worsen their case, they make treatment difficult because the drugs available for those neurological disorders may not be sufficient to actually reverse the problems that will be caused by those exposure, those toxicants. So a call to our scientists in Africa is that our research to be localized. And aside from that, we should collaborate widely. And it is evidence from our group that we do not have everything. We cannot boast of big funding, right? But we can boast of good collaboration, right? So even with minimal resources, I think that we can do well in Africa, right, to resolve the complex neurological puzzles. There's what we call neglected neurological disorders in Africa, caused by smoke from firewood, caused by so many things that we have not studied yet. So in acknowledgement, because whatever you see that is good is actually a contribution or collaboration with some people. I appreciate the platform provided by SONA once again, and then the opportunity given to me to be in an academic environment that I've a training, and then also it can give me opportunity to visit some labs or interact with some mentors, and then my mentors, right? So these are the big guys that actually kind of motivate me. I see them, then I also wanted to work. Then I have a list of collaborators, more specifically, right? Dr. Jomane, right? So he has provided a lot of assistance in this work. So I think the end statement is that we are open to more collaborations, and then we'll appreciate what we call you when you feel what we're doing is so thank you so much for your time. I think I've spent my 40 minutes. Right?
Yeah, certainly. And thank you so much for such an insightful presentation, Dr. Imam. It's great to see the level of interaction that this has generated, of course, in the chat box among the viewers. And we thank all viewers for staying tuned, for finding time to join this, and for typing in your feedback, a lot of which has indicated positivity, which we are glad to hear. And some people have also been kind enough to drop their questions as well, and we'll dive right into them. It's of course nice to see people joining from all over. This is quite impressive. Thank you so much. So to start with is talking about adult neurogenesis. So Bismarck Apia is wondering if you consider the effects on adult neurogenesis and subsequent implications on memory. What would you say to that, Dr. Imam?
Can I get the question again?
Yeah, so it's projected on the screen. So he's wondering what effects we could have on adult neurogenesis. Yes.
So thank you so much. I think just as we all know that there are hotspots within the brain, and these hotspots majorly are actually in the subventricular zones, right, or kind of related to the hippocampus, or even subventricular zones in the olfactory bulb and the rest, right? We all know that these cells, they kind of retain their integrity in these regions, right, until adulthood, as many reporters have kind of said, right, and then they can be recruited when there's need. In fact, we can say from this point that if there's gliogenesis, right? So if there is gliogenesis, right, definitely this structure or this toxicant will have effect on neurogenesis. So it will have effect on gliogenesis. And the reason I'm saying this, for those in the glial world, they know what to call the radial glial cells. And this is actually the source of every other cells that we see. So I think I've not studied it myself. I've not been interested in immunogenesis in the recent time, right? But I've read it somewhere where it says that, right? So antioxidants actually encourage neurogenesis, right? How that happens, right? Maybe through preventing the kind of they can through encouragement of nootropic factors, right? Encouraging the production of nootropic factors and right. But specifically, if the insecticide we use here affect neurogenesis, right? In comorbidity, we've not studied that. And I think it should, if it affects gliogenesis. And people understand when I say if it affects gliogenesis, it's well, definitely affect because neurons, just like people have tried to correct us over the time, but neurons do not exist in the brain alone, right? When we say neurons fire, we just say because they're the major cells that do that. Glial cells are central to both function of neurons, dysfunction of neurons, maintenance of neurons, health of neurons. So, and I think the brain is so complex that it has both gliogenesis maintained in adulthood and neurogenesis also maintained in adulthood. So I think if it affects gliogenesis, it will affect neurogenesis. And yeah,
and then we'll keep to that.
For memory, we already said that before. Right, thank you so much.
Yeah, High Faith is wondering what extent vitamin E attenuated the effects of ETZ on MBP, GFAP, IBO1 in terms of the quantitation.
Yeah. Thank you so much. I think when I shared my PowerPoint with one of my mentors, what he said was to include the quantification, right? But from our published papers, we found quantifying the cells, right? We saw that just for presentation sake that I brought, and just so that people can see clearly, right? Histochemistry, right? Not to kind of burden our eyes with just figures flying around, right? So we saw that both immunoactivity, that's when you check for the expression of protein, and the counting of the cell, that these cells actually there is reduction in myelin-based protein expression, right? And also the population of the GFAP positive astrocytes and ADO1 positive microglia actually increase significantly. And like I said, where we do not see any significant results is in cerebellum, right? We don't know why, right? So, and then probably we'll actually kind of, it's still an ongoing research, so we'll kind of look into it. Probably we have not covered enough, so probably we'll have to remodel and then check that region again.
Okay, then. And this moves us to a question about the timeline of remyelination from Demigovenda. How long after toxicant exposure did you see signs of remyelination?
Okay, so thank you. Right, so one of the things that those who work on myelinating research actually can avoid is what we call physiological remyelination. So immediately after demyelination, there will be remyelination. In fact, this is one of the devil in modeling demyelination, right? Not following standard procedure, because you may end up having your standard control, standard model remyelinated before you're ready, okay, or whatever. But what we compare our study with is that we terminated the experiment immediately, right? We terminated the experiment immediately, and that way we're able to know that vitamin E that we use to supplement nucleic acid prevented demyelination in the first instance, right? Prevented demyelination in the first instance. Because we know that there's physiological demyelination. How much a substance encourages demyelination is something that needs your attention, actually, to understand the drug very well, to understand the model very well. So for us, immediately after the exposure, right, for about 4 weeks, we stop the experiment and then we know that there's demyelination. Right. Why in the group we need remyelination to occur? We expected, we hypothesized remyelination. We gave the drugs to prevent demyelination, right? And then because we know that even while we give the drug before exposing to cypermethrin or PTZ, we know that the drugs may not have started working. Right? So this is why we call it remyelination, right? This is why we call it remyelination, because the structures that support myelin integrity are actually healthy as far as our study is concerned. But about the timeline, you have to be careful when you induce even demyelination with the standard clinical models. You have to either start the treatment immediately or you terminate your experiment immediately, but there's remyelination normally. But they may not be complete remyelination. They will not be complete remyelination. So what we see is just most of these are just partial demyelination and remyelination.
Thanks. I think that's a good place to leave it. You've commented severally about the demyelination that is not observed in the cerebellum. Do you have a hypothesis as to why?
So we don't know why. Just like I said, so the part of kind of looking at the whole brain is actually still ongoing, right? I may just say that in fact many studies have reported that there is demyelination in the cerebellum, right? Right. So especially the Enigma study actually shows throughout the brain cerebral white matter. Right. So, but we did not see it in our study. Like probably when we repeat it, the work is still ongoing. But we don't know why. In fact, we think our thought was that would be the first point of demyelination. But since we didn't see that, so we don't report what we did not see.
Yeah, some literature may actually be helpful in that sphere, but I think we can come back to that with updates from your lab, actually. The next question is about the mechanism of demyelination. So if one nerve fiber is demyelinated, Samuel is wondering if it can spread or affect other nerve fiber. What do you think, Dr. Imam?
Okay. Thank you so much. That's a very strong question. I've never thought of that, except if the demyelination is kind of caused by an infection, right? But that a demyelination, demyelinating X-ray will cause the other. Okay, there's what we call induction in biology. So basically, I do not have a straightforward answer for that. I don't know, right? And I'm sorry to say that.
You don't have to be sorry. That's what the community does. Thank you so much for leaving it at that point. I'm sure even Samuel is further inspired by your answer. Perhaps he could pick that up better for the investigation. Now, Remya is thinking that there could be a connection between the efficacy or efficiency and the route of administration. Which one did you use? Was it IV or oral?
Okay, thank you so much. Yeah, we use oral, and there's a reason for that. Most antiepileptic drugs, except in emergency case, we use it can be taken through oral route. So if we could not see any result oral route, most times when we study using other route, it's just to understand the mechanism. Right? To kind of get the clinical advantage of this, right? We think using the oral route is kind of may be wrong, right? So maybe the best. Because when epileptic patients use their drugs, they take it orally. So if the drugs cannot work orally, right? They cannot work orally. So that's just my own judgment, right? I may be wrong. Many people have used different routes, right? And many will argue that if you want to get a faster mechanistic response, right? You use probably intraperitoneal, subcutaneous, or any other route, right? But we chose oral route because the drugs that we use actually via oral route. So if they cannot work for the time we will just say that they cannot work for the time. Right, then they kind of either increase the treatment or whatever. So that was all we did. We use orals. Right. Right. So people have said, just like we all
That's
learn,
a nice thought to
just like
have.
we all learn in pharmacology that there's first
Mm,
pass mechanism. Right. So there's first pass mechanism. Right. So I
mm.
know that too. Right? But the drugs we take, once they can cross the blood-brain barrier, right, they will definitely cross. The amount that cross,
Thank
most
you.
times.
Mm.
Okay. Yeah, so we at this point, maybe while we still see, we'll have more questions from there. I was just wondering, since you mentioned the particular vulnerability of oligodendrocytes to oxidative stress. So do you think targeting the oxidative pathways early in epileptogenesis could have a meaningful downstream effect on white matter degeneration?
Yeah. Thank you so much. I really appreciate this. I did not think about this until about, I think, a month ago, when an undergraduate student was actually presenting on remyelination and demyelination. And then like four of them, actually opened my eyes to more mechanism, right, rather than what I'm interested in. I now realize that even when we say that oligodendrocytes, because oligodendrocytes, astrocytes have what we call connection, right? And oligodendrocytes for themselves, right, could die. They may also, just like they are also cells, right? Right. See, they are cells.
Yeah.
Oxidative events in them, right? And inflammatory events in them, right, is what leads to most time they're dead. Right.
Mhm.
So we think if you prevent oxidative stress, right, in epilepsy early, right? So if an epileptic patient take probably a flu or take supplements, antihistamine supplement, right? And the problem in that patient does not exceed regulatory inhibitory value. That
Okay.
is actually introducing antioxidants that control
Mhm.
receptivity. That it will be very helpful rather than waiting for the first episode, then use your drug. Right. But
I see.
this is not clinical. However, there is actually a clinical study, right? A
Mhm.
clinical trial that has said that vitamin E, right,
Mm.
is anti-epileptic, right? And the mechanism is
I was actually coming to that too, but that was thank you very much. Yeah. So as I see that we are just about the top of the hour, one interesting your presentation is the neurological risks that are associated with pesticide exposure. Pesticides are everywhere, and we know how much it means to many African agricultural communities, right? So I think it would be nice you give a comment on environmental toxicology, perhaps as a frontier for neuroscience research on the continent. Maybe that will be limited to sub-Saharan Africa. I don't know, but I would like you to just say something at this point about that because you are in a good position to do that.
Thank you so much. I think I may both to just say that we should all just be toxicologists, right? Because in the recent of this, right, we know that we have a tradition of dependence on non-standard drugs, right? All Africans know that. I will take one plant, we make concoction out of it, and we will take it. And there's a theory in toxicology that is usually in kind of a general thing that says the dose is the problem. Right? The dose is the problem. Most of the things we take do not have dosage. Right? We just take them. Right? So, and just like we take every other thing, you see in fact there's a news in Benin where a family, like of six, slept in a room with insecticide in the room. And they woke up in the morning, half of them are unconscious, the mother is dead. right? So this is because when we use things in this part of the world, right, we use it excessively, right, and indiscriminately because we want results very quick. Just like farmers will use too much of insecticide, we
Yeah.
also use too much of insecticide. I think neuroscientists in Africa, right, should study more these chemicals. And I've seen an example in Morocco a long time ago when they work on paraquat and the Parkinsonism, and they did that for a long time. But I think we should all invest in that. So if you are in, for example, Alzheimer's disease, if your model of interest is Alzheimer's disease, you can as well comorbid that's your model with organophosphate, which are cholinergic disruptors. Right.
Yeah.
If you're somebody who is interested in Parkinsonism, you can as well comorbid that model with another insecticide, or for example, paraquat. Right. So
Mm.
in this way, we use that to study our environment and then to provide advice to the community that when you use this insecticide, what
Yeah.
it did to this brain is what it can do to your brain, right? And
Exactly.
then, but we also need environmental and public health studies, right? Rather than just experimental studies, right? We also
Certainly.
need.
Yeah.
You'll be surprised that if you go to the literature, most of
Mm.
these results they are just at our arm's grip and not
Mm.
available. Like this. So, and this
Exactly.
is where we kind of collaborate, right? Make that out of different countries and then make it available to the news outlets, kind of to
Mm.
advise.
That's all round. And on that note, I really want to thank Dr. Imam Aminu as we come to the end of today's session, all the way from the University of Ilorin in
Okay.
Nigeria. We certainly hope for future updates from the Neurotox group. All
Yeah.
right, Dr. Imam. So we thank you
Thank,
so much. We hope you're going to yield our call the next time we ask for updates from your group. And for the
thank you so
audience,
much.
thank you so much for everywhere you're joining. There has been a lot of nice feedbacks in the comment section. Thank you all for staying tuned, for finding the time, for dropping your comments, for feeding us back. We appreciate that. And like I said earlier, there are going to be feedback forms at the end after this webinar, and those that are interested in certificates of participation would also be attended to. So thank you all for joining from different parts of Africa. Please stay tuned for future SONA webinars. Like we said, stay tuned for more scientific content as well as career development sessions. On this note, I'm happy to close with a quote from Santiago Ramoni Cajal, as we call him. Yeah, not Cajal, right? So he says that every man, if he so desires, can become the sculptor of his own brain. So that is Cajal referring to neuroplasticity, part of what Dr. Imam's work on Glia also alluded to, and as we have from one of the questioners. But then if this has to do with how each man's experience influences how the brain comes out to be, we are saying this is a perfect avenue for us all to also come together to continue shaping neuroscience in Africa. On this note, thank you all for joining us. We look forward to seeing you at the next SONA webinar, and we wish everyone a good day or a good evening, depending on where you're joining from. At this point, we say bye until we see you next time. Bye, everyone.
Thank you so much.